welcome to this. I'm Nick Stewart. I've been doing a number of webinars for BSBI on aquatic plant identification. And one of the questions that comes up quite regularly is how best to preserve aquatic plants. So I'm hoping just a short discussion about the main things, the main ways of doing so. There are two main ways of preserving aquatic plants. There's pressing, which is by far the most common way of doing it, or pickling in alcohol, and I'll discuss both of these, the pros and cons of both. But the vast majority of things is a way of preserving things is by pressing them, and this I'll tackle this first. One of the problems with pressing things is aquatic plants is that they often have rather low rigidity, so it does mean that they tend to clump together, and you could spend quite a lot of time just sort of gradually pulling apart the different leaves to lay it out, but that's quite a time-consuming process, and becomes even more difficult if you've got some of the feathery leaf things like water milfoils and water crowfoot. And there is a much easier way of doing it, which is to float them out. So what you would do is you would get your specimen in a tray in a little bit of water, and you then put a sheet of paper, I tend to use just A4 paper, and slip it underneath the specimen, still in the water. So the water, so you have the sheet of paper under the water, and then you lay out the specimen to fit it onto the piece of paper and how you want it, and the water will keep the leaves separate, so they don't clump together in the same way that if you just pull it out of the water. And then very carefully you lift the paper and the specimen out of the water, so that the water just gently drains away. If you do it too quickly, then you'll get a wash of water and it will pull the specimen with it. But if you do it very gently, and just pull it out of the water, you'll find that it will stick very nicely to the paper. And then you just let the excess water drain off and put it in the newspaper. And you see that with very little effort, you have a much easier, well laid out specimen that you can see all the features very well. Now, what you'll find is that specimen will stick to that piece of paper, and that piece of paper will actually then become part of the specimen. But the problem is, if you then just close up the newspaper and the press, it will stick just as easily to the newspaper on top. So the trick for this is to get some baking parchment, and if you lay that over the top, you'll find that it won't stick to that. It's the waxing in the paper that stops it sticking to that. And you'll find that when the specimen is dry, you just need to open it out and take away the cooking parchment, and you're just left with the specimen sitting on the piece of paper that you pulled it out of the water on. If you don't, you'll find that when it's dry, you'll end up trying to sort of gradually pull away the top piece of paper, and often you'll damage the tips of the leaves, or they'll get caught on the wrong side of the bit of paper. So this is actually a really useful tip. The only alternative to doing that is that if you disturb it regularly during the drying process, so before it's completely dry, you open it up. You can then make sure that it's not sticking to the paper. But this is a much easier way of doing it. You can just put that over, and makes your job considerably easier. So then you just close it up and let it dry in the normal way that you would do with any sort of specimen. Probably if the specimen was very wet, then you might need to change the paper once, sort of after a day or so. But often, because the specimens are quite small, they will dry out quite quickly, and probably in a day or so if you've got sort of dry papers on either side. Then when it's dry, as I say, what you would do is mount the specimen on the paper that you had it, and mount that onto a herbarium sheet if you're making a herbarium specimen. And if need be, you'll find that most of the specimen is stuck to the paper, but you might need a little bit of glue to just sort of stick down any loose bits that are that are waving around. Also, make sure that the specimen's labelled. The key things to have in the labels is just as in all herbarium specimens, you need to have the species, locality, who collected it, and the date are the key bits of information, but any extra information is always useful. And you'll find that it's really quite easy to do, and it will only take you a few minutes to just float them out, even though it's just a little bit more complicated. Some additional points to say: if you have access to acid-free paper, that's better for long-term storage than sort of herbariums. And often, if you have contacts with herbarium, they will often provide you with some of the materials, such as acid-free paper, if they know that they're going to accumulate specimens for the herbarium collections. So it's worth talking to any herbarium that you might be giving the specimens to in the future. Only preserve one specimen, one species from one locality on each sheet. If you have multiple species, then it complicates. That tends to be how they're stored in herbaria. And if you have multiple species, then people don't know which species to file it under. So best thing is one species from one locality on each sheet. In general, because aquatic plants people have had more difficulty with, it is worth keeping batches of material on a fairly regular basis, but particularly if it's a new record for the site, or if it's a particularly critical species, it's worth keeping a batch. But you do need to be aware that some species are especially protected and require a license. They're different species in the Republic of Ireland and Northern Ireland and in Britain. So I won't try and list those here, but things like the Greenlandia, which we were discussing earlier, is specially protected in Ireland and shouldn't be collected without a license. Also be careful if there only seems to be a small amount present, then again, I would say don't collect material. But on the whole, the very method of sampling involves taking the plant out of the water to actually look at it, to identify it. So you've already damaged the plant just in the process of that. And in most cases, you'll find that aquatic plants occur in quite large stands, and it's no particular issue to collect material and to keep a bit of that if you've already got a bit out to identify. The other way of preserving things is to preserve in alcohol. Generally avoid other chemicals like formalin and formaldehyde, and pretty nasty and carcinogenic. But so most preservation, liquid preservation, is in alcohol. There are a lot of disadvantages in preserving in alcohol. It's bulky. You can very easily build up quite a large cupboard full of the specimens if you're preserving them in alcohol all the time. They need periodic maintenance, topping up to make sure that there aren't cracks in the collecting tubes or just evaporation. So that they do require maintenance. They do lose colour, and often there will be destruction of any genetic material if you're. Whereas you can get genetic material from well-dried herbarium specimens certainly in some alcohols. It destroys the genetic material. There are also problems sending it through the post. There are issues sending inflammable liquids, and that can be quite a problem. So why do it? There are certain advantages. It's very quick to do. So in fact, a lot of the specimens that I get that are pickled in alcohol, it's people that are doing a lot of field work day on day, and they have rather limited time in the evening to process any specimens that they've collected. So it's very quick just to stick a few sprigs or something in a tube with alcohol and put a label on it. As I say, that's probably one of the main advantages of it. With calcified samples, they tend to be less fragile, and there are some characters that are preserved better in liquid preservation, particularly flower shape, and also measurements are preserved better. In dry material, things can shrink, and you're not getting a true measurement. Particular areas where people tend to use liquid preservation a bit more, there are sort of three genera, I would say, particularly that liquid preservation is used. Duckweeds don't tend to press very well, particularly if you've got sort of three-dimensional things like the fat duckweed Lemna gibber. If you press that, then it loses all its sponginess. So that's one area where there's can be an advantage in preserving in liquid. Bladderworts is another group which are quite often preserved in liquid. The two parts to that. The flower shape is quite useful as an identification of things, particularly between Utriculata gargaris and Utriculata australis. And it's to do with the shape of the lower lip. And of course, if you press that, that gets completely distorted. Also in bladderworts, an important character for separating, particularly in the intermediate group, are the hairs inside the bladder. Inside the bladders of bladderworts, there are these little four-armed hairs, which help to dissolve the little beastie that the traps have caught and digest them. And they are quite a useful identification character, particularly in the intermediate group, because there are very few flowers in the intermediate group, very rare flowering. And those hairs are quite fragile, and when they're pressed, they become impossible to actually rehydrate and see the hairs. So very often bladderworts are preserved in alcohol. Another group where preserving in alcohol is sometimes advocated. I personally prefer dried material, but I do get quite a lot of pickled material. And there are some advantages. Some of the characters of the cortex, the stripes in the stem are easier to see in material that's preserved in alcohol. Also, the size of the fruiting structures reduces quite considerably when they're dried. So if you're taking measurements, but even so, there are, you can see those characters in dried material, albeit in sort of appearing in a different way. So pressing material is perfectly reasonable for stone wets, and again, the advantages are that it's a lot easier to maintain the specimens, to store the specimens afterwards. But it is a valid way of preserving stone wets. Some just general points. It's wise to use containers with straight necks. It means that when you're pulling material in and out to look at them, it's a lot easier. If there's a constriction at the neck, then it can be quite difficult to get the material out of the things without damaging it. And also very important is that they have good seals, because otherwise the things will the alcohol will evaporate very rapidly, and you're just left with a dry crumbly specimen. So common containers are things like sterilins, they're called, these tubes with very good seals, sort of things that you would do urine samples for the doctor in, and they make very good containers for storing aquatic plants. The preservatives, recommendation is 70% alcohol. You can, it doesn't need to be quite this strong, but that's the general recommendation. And the most often used is industrial methylated spirits, IMS, which is a lot easier to get hold of than ethanol. Ethanol has all sorts of licensing things, so you're unlikely to get hold of it easily unless you're attached to an institution. But if you're working on genetic material, it does preserve the genetics better. You can use gin, vodka, or any clear alcoholic drinks, although the percentage alcohol tends to be a bit lower, so it probably isn't so good for the long term, but certainly they're very effective for initial preservation until you can get stronger material or from a herbarium or some such. I've never tried it, but some of the antiviral hand disinfectants, which have become commonplace due to COVID, if they're very liquid versions, the percentage alcohol needs to be 70% to kill the COVID virus. So theoretically, they may be possible preservatives for aquatic plants. I've never tried it. I don't know if there are other things that they put in which wouldn't be so good for the plants, but it's another possibility. You can add a little bit of glycerol. It helps to keep the specimen a bit more flexible, but most of the time people are not doing that. But if you're collecting particularly for a herbarium, they will often add a little bit of glycerol just to keep things a bit more flexible. A general tip is best not to overstuff the container. It is very tempting if you've got a lot of material and you want to sort of some nice bits to preserve, to actually put too much. The problem with that is that when you're actually looking at the specimen afterwards, sort of taking it out, you have to do quite a lot of entangling to look at the specimen, and that can cause the damage if they're all sort of wound up in a knot. So something a bit looser it tends to be much more preferable. And you can also see here that one of the disadvantages of pickled material is that it loses all its color. And you're just left with a sort of rather grayish plant. But you can still see the features, but the color back, it gets completely lost. Just some points about labeling. It's important to have permanent ink or pencil on the labels, and it's quite a useful fail-safe to actually put a label inside the container, just in case the ink gets dissolved away. Some permanent inks do get dissolved by the alcohol, so you need to be quite careful that you don't lose the labeling information.